Hello, everyone. Knowing the fact that membranes have an important role to play in transport across, in this session we would look at the principles of membrane transport or the principles on which the transport of molecules across the membrane is based. So let us look at the learning outcomes of the session. Membrane transport is defined by the fact that membrane is semi-permeable. That means that it is only selectively permeable to certain molecules. And the reason for this is that when you look at the structure of the membrane being a bilipid layer, the inner environment is hydrophobic. So therefore, when you look at transport across the membrane, you will find that that can be a deterrent to the transport of a molecule that is hydrophilic. So therefore, you immediately kind of are able to sense that because of the structure of the membrane, you have permeability or you have semi-permeability associated with it. So it allows water, small molecules to pass through, but not large solutes and ions. Based on the principles of diffusion and osmosis, there is transport of molecules across, which in itself is very important for the cell because that helps in maintaining the osmolarity of the cell itself. The various transport mechanisms comprise simple diffusion, facilitated diffusion, and active transport. And transport across can also be by vesicular transport through endocytosis and exocytosis. So let us look at what is the principle of diffusion, or basically understand the principle of diffusion, which was given by Fick. And these laws are called as Fick's laws of diffusion based on the simple fact that there is a tendency for solutes to move from a region of higher concentration to one of lower concentration. That means they are able to move down the gradient, not up against the gradient, but down the gradient, and that movement is spontaneous. So let's say, for example, this is a semi-permeable membrane, and when we consider permeability, let us consider it that the molecules here will be shown will be having low permeability, and those that are shown up will be having high permeability. And in between, you would have the permeability varying based on where it is existing. So, for example, water, carbon dioxide, oxygen, etc. have very high permeability. They can simply diffuse through the semi-permeable membrane. Urea glycerol has permeability but lesser than that of water, carbon dioxide, and oxygen. Tryptophan glucose can also diffuse across the semi-permeable membrane, but the permeability is less. And the least permeability is that of ions. Whether you take chloride ions, potassium ions, or sodium ions, all the three have the least permeability when you consider a semi-permeable membrane. So what is the observation that you kind of make from this? One, that if they are large solutes and if they are charged, because you see they are ions, then they diffuse slowly across the membrane. But if they are uncharged and small molecules, then they can diffuse across the membrane easily. So just to look at that simple concept of osmosis and simple diffusion. So when you look at osmosis, say for example, a simple experiment that was carried out in a U-tube, and in the U-tube, at this position, a semi-permeable membrane is added. And suppose this arm of the tube is filled with a solution of sugar in which the sugar concentration is high, and in this you have the same sugar solution, but say for example here the concentration of the sugar is less, or consider that these are any solutes, but here the concentration of the solutes is high and here the concentration of the solute is less. So what is observed is the phenomenon of osmosis. So what will happen is, because you have a high solute concentration in this arm, water from this arm will move into or diffuse into this arm. This is because the water can move from So for water, the amount of water here with respect to the solute concentration is higher. Or you can say that the solute concentration being less over here and the solute concentration being high over here, the water moves from a region where there is low solute concentration to a region where there is high solute concentration. So you can see how the level of water has become higher in this arm because the solute concentration is greater. So this is what we understand by osmosis, and this is exactly what really happens in cells. That is, water can move across depending on where the solute concentration is high or where the solute concentration is low. Now, if you want to push back this water, then you will have to have a pump that puts pressure, and an equivalent pressure that is needed to pump the water back to this is what is understood by osmotic pressure. So let's consider, for example, you have an RBC, that is a red blood cell, and a red blood cell is kept in an isotonic solution. So when you say isotonic solution, what does it mean? It means that the solute concentration inside the RBC is almost equivalent to the solute concentration present outside. So that is what is called as isotonicity. In such a case, you would only have the water going out and in, but maintaining an equilibrium. However, when you, for example, put the RBC in a solution that is hypertonic, which means that the solute concentration present outside is higher. If that is the case, then water from inside the RBC is going to move out because of osmosis. And so what is going to happen to the RBC is that the RBC will get crenated or shrunk. Okay, so this is definitely detrimental. And the other situation is that if you put the RBC in a hypotonic solution, so what does that mean? It means that outside the solute concentration is less, while inside the solute concentration is higher. So water from outside will start entering the RBC and the RBC will become swollen. It will get swollen to a point that would make the RBC lyse. So therefore, this actually is used to lyse the RBCs if one wants to. That is, you put the RBCs in a hypotonic solution and immediately you will find that it will lyse. So therefore, this is what is happening in real cells. So when you consider that the blood cells have to move around in blood, it is important that the blood uh, has a concentration of solute outside which is almost equivalent to the concentration of solute inside. So this is something that is what we understand by osmosis and the effects of osmosis, and the fact that the every cell also has an osmolarity of its own. When you say osmolarity, it means that the concentration of solute that would help maintain a particular osmotic condition. Now we look at simple diffusion. So when you have a plasma membrane, we know that it is a bilipid layer, and the bilipid layer is made up of amphipathic cells, amphipathic molecules. So within the bilipid layer, that is this bilipid layer, this portion is completely hydrophobic. So therefore, the hydrophobicity is making the membrane less permeable. Now, the heads that are present are definitely something that are contributing to making the membrane semi-permeable. That is definitely the case. Now, the other thing also is that you have these phospholipids or you have these lipid molecules or proteins, etc., in this spaced out. So there is a certain amount of spatiation present within these molecules, and therefore that calls in for a certain level of permeability. What has been observed is if you have gases, gases can freely move across the membrane. Okay, so if you have higher concentration of oxygen here compared to outside, then oxygen will move out. So the diffusion will always be towards the towards the side where the concentration of the molecule is greater. Where it is lower. So it will move from a higher concentration to a lower concentration. So that is what a gradient is. So this is what is a concentration gradient. Now, when you consider benzene, etc., which are hydrophobic molecules, then because you have the bilipid layer being hydrophobic, you will find that a benzene will easily move across the membrane. So is the case with sterols. They also will move across because sterols also are hydrophobic. Now, say for example, you take small polar molecules. So far we considered gases and we considered hydrophobic molecules. Hydrophobic molecules can easily pass through because the membrane per se has a lot of hydrophobicity. Now, say we consider small molecules like water and ethanol. Ethanol is amphipathic. So therefore, its small size and the fact that both of these are uncharged, no charge on them, they're able to move across. So for all these three, there is absolutely no involvement of membrane proteins in their transports. And that is the reason why it is called a simple diffusion. Now, you must understand that water over here is polar, but it has two advantages. One, that it is small in size, and secondly, it does not have a net charge on it. And therefore, what you will find is that these water molecules are able to traverse across. Even if there are kinks, etc., present in the bilipid layer, you still will have the water move across. So, it is highly permeable. So, water is easily permeable through the membrane structures. So is the case with ethanol. And what will happen is that you would have a constant flow in and flow out of the molecule happening, but it will reach an equilibrium where the concentration inside and outside is the same. Now, say for example, you go in for slightly larger polar molecules, say for example, glucose. So, the glucose being slightly larger compared to water and ethanol and also being hydrophilic, okay, it cannot very easily permeate through. So, therefore, you can immediately see that the permeability for glucose is much lesser than the permeability for this, and so it is not very easy for the glucose to diffuse through through its own. Same is the case with amino acids and ions. The ions are charged molecules, so and they would have large layers of hydration around it, because of which the size of these ions will also become large, and so these also will not be able to move across on their own. But steroid hormones, as mentioned earlier, because they are hydrophobic in nature, they can pass through the membrane. So you can already see how through simple diffusion without the aid of any membrane protein, you can have molecules moving across. But the catch is that they are going to move across dependent on the concentration gradient across the membrane. They would move from a higher concentration to a lower concentration. Now we come to another mode of transport, and that is facilitated diffusion. So say for example, you have a semi-permeable membrane, and outside of the membrane you have a large number of glucose molecules present. Now these glucose molecules, to permeate through the membrane on its own, it is very difficult. So it is not easily permeable because of its molecular weight. Now, however, if there is a protein present and this protein forms or is able to somehow carry the molecule across, then what you will find is that you will have the sugar moving from a higher concentration to lower concentration. So here it is a process that is membrane enabled, and that's the reason why it is called as facilitated. It is diffusion because it is still moving from high concentration to lower concentration, but then it is being aided, it is being helped, and that help is given by, say for example, a membrane protein. Then this type of a transport is what is understood to be facilitated diffusion. Now we go to another facilitated diffusion, which is called as active transport. So say for example, you have a membrane and then you have a transporter, but say for example, this transporter has to transport this molecule across to the outside. What you can observe over here is that the concentration of this molecule outside is already high, and yet you have to kind of push out the uh, molecules to the outside itself. Then it means that you have to move the molecule across the membrane but against the gradient. So ideally you would have the molecule, because the concentration is high over here, you would have the molecule move to the inner side. But then this transporter has to transport the molecule to the outside against the concentration gradient. So here you have higher concentration, here you have lower concentration, but the transport has to be against the gradient. So for that, a membrane protein is definitely needed, and that membrane protein uses an ATP to pump the molecule across the membrane. So what has been observed is many of these membrane proteins that carry out active transport have an inherent ATPase activity, which means that they are not just structural proteins, but they are also enzymes, and they are specifically able to use ATP and hydrolyze it to ADP. So from ATP to ADP, energy that is released, that energy is used to pump out the molecules to the outside, because that has to be against the concentration gradient. Now, looking at all these different ways of transport, there are three main types of transport that happens across the membrane. One is what is called as the uniporter. Now, this can be facilitated diffusion itself. Okay. Now, uniporter is basically when you have the transporter transporting only one molecule. So, it can specifically transport only molecule A. Now, the transportation can happen according to the concentration gradient. If the concentration of A outside is greater, it will move in. But if the concentration of A inside is greater, then it is going to move out. So this will happen until it reaches an equilibrium across the membrane. If you have the same amount present inside and outside, you may have the molecule moving out, but then it is only moving out, in maintaining an equilibrium. So it is constantly moving, but it is just maintaining an equilibrium. So that is when you have a single molecule being transported across through one particular protein. Suppose you have co-transportation. That means that one transporter is enabling the transport of not just one molecule, but transport of two molecules. Then if it is able to transport molecules towards one side, so both A and B are transported in the same direction, then that is what is called a symporter. So one of the best examples for this is the sodium glucose transport that is present in the intestine. So we all know that intestine is where absorption of food takes place. So the intestine has a large number of sodium glucose transporter, these glucose transporters basically, and these glucose transporters will bind to sodium and will bind to glucose, and together the sodium and glucose will be able to be transported inside the lumen of the inside the intestinal cell. So that is how the symporters function. Then you can have antiporter. Antiporter is again co-transportation, but the transportation of the two molecules is in opposite direction to each other. So for example, if A is being brought in, then B is being thrown out of the cell, considering that this is intracellular and this is extracellular. So here you have the movement of two molecules across the transporter, but the movement is in opposite direction. And a very good example of antiporters is sodium potassium pump. So we all know that all our cells, or many cells, have sodium potassium pumps that are able to maintain the potential across the membrane. So you see how transportation across the membrane is also enabling maintaining a membrane potential, a resting membrane potential, and if one moves away from this resting membrane potential, then it can give rise to an action potential. So sodium potassium transporters have very typically three molecules of sodium being thrown out, while two molecules of potassium being brought in. So uh, so interesting to note is that in case of sodium potassium pumps, it is moving against the concentration gradient. So sodium being pushed out is against a concentration gradient, and potassium being pushed in is again against a concentration gradient. So sodium potassium is therefore called a pump, because it needs ATP to push the molecules across in opposite directions, and not just opposite direction, against the concentration gradient. Now we look at another transport mediation, and that is through vesicles. Say for example you have the membrane and this is a molecule that has to be transported across. Then what is observed is that the membrane is able to curve to form an invagination. Okay, and this curving of the membrane could be because of the membrane lipids itself, and also because of the proteins coming and binding to these points where it can curve. Okay, so once this has invagination has formed, what happens is the ends of the invaginated structure would come together and fuse with each other, and at this point it would be pinched off. So now you have inside the cell a vesicle containing the molecule that needed to be transported. So therefore it is called as vesicular mediated transport, and this process is what is called as endocytosis. That means the cell is taking up something inside. Now, what has been observed in many cases is also that the cell membrane may have receptors present on it, and to these receptors you will have the ligands binding. So once the ligands are bounded to the receptor, then at this point you can have invagination formed. The receptor along with the transporters are then pinched off as a vesicle, and now inside you have the receptor with the ligand present. So this kind of an endocytosis is termed as receptor mediated endocytosis. The opposite is exocytosis. So in that case, a vesicle which contains a molecule inside will fuse with the membrane, and on fusing the membranes, it will invaginate out, and then you would have the vesicle moving out. Sometimes what can also happen is that simply you have this opening up and this molecule being released. So you don't have it going as a vesicle, only that the molecule is thrown out. But that also is what is considered to be exocytosis itself. So this is how you have transport across, but this time what you are very clearly understanding is that it is going through in the form of vesicles. So let us make the conclusions. Transport across the membrane is based on the principle of movement of molecules across a semi-permeable membrane down the concentration gradient. Simple diffusion is molecules moving across without the help of any protein in the direction of the concentration gradient. This is always the case. When you look at diffusion, it is always in the direction of the concentration gradient. Facilitated diffusion are two types. One that needs proteins. So we are talking about facilitated diffusion where just a protein is needed but no energy is needed. And the other which we are talking about is active transport, which needs both the protein and energy. Transport across can also be mediated through vesicles, which is called as endocytosis and exocytosis. So membrane transport is dependent on the size and the concentration of molecule to be transported. They can be transported as a single molecule or they could be co-transported. They can be transported against the concentration gradient as well. Thank you.